Hello students, welcome to EPG Pathshala. I am Dr. Mini Singh from Department of Biotechnology of Punjabi University, Patiala. Today we are going to discuss about food nanotechnology and its introduction under the paper Nanobiotechnology. During the course of this entire module, you have studied about the basic biomolecules, their nanostructures. You've also studied about various applications ranging from pharmaceutical applications, environmental applications. But one lesser known application of nanotechnology is in the food sector. That is why it is important for us to understand what nanotechnology holds in this sector also. This module has been specifically designed to give you an introduction of the various applications that nanotechnology holds in the food industry. The reason why this is important is because nanotechnology is gaining impetus in various fields, and one important field is in food applications. How this happens, what are the various applications of nanotechnology in food will be discussed in this module. These 6 modules are the last modules of the paper on nanobiotechnology, and we will take you through certain specific examples or illustrations too during the course of this paper. Based on this module has been designed with the following objectives: nanotechnology in food applications, wherein we will discuss 3 separate applications. First is the food safety nanobiosensors, followed by nano formulations in food applications. Nano formulations are used for enhancing the value of certain foods for human use, followed by nanotechnology in food packages. Now, food packages is a very interesting application of food nanotechnology, wherein we will see how nanomaterials have been used or impregnated or devised inside the conventional nanomaterials, and how the properties of these conventional packages enhance. When we say properties, they could be tensile strength properties, they could be mechanical properties, they could also be barrier properties. With barrier, we mean whether the impregnation of these nanoparticles within the conventional food packages is able to enhance or diminish the barrier of certain gases. Where do these gases come from? These gases, since we're talking about live food materials, when live food materials are stored, there is respiratory activity. When respiratory activity happens, there is exudation of certain gases, and those gases become a marker for telling us whether the food is still safe to consume or whether it is in a position where we have to withhold the production or sale of that food package. Despite these opportunities which food nanotechnology holds, the various applications that we will be understanding, we understand them as opportunities which are taken up by scientists and using these particles or nanomaterials for certain applications. However, we also have to understand what are the challenges that nanotechnology poses towards the use of these materials or the applications of these materials in the food sector. Coming to applications of nanotechnology in the food sector, these are broadly classified into 2 types. One, which is the out-of-food applications, and one, which is the in-food applications. When we talk about in-food applications, we are essentially talking about those applications which involve direct contact of the food material with the nanoparticles, or fabricating particles in such a manner that they themselves become a part of the food. In-food applications also means that we are talking about formulations, we are talking about sensors which come in direct contact with the food, or we're also talking about nano additives. Whereas the out-of-food applications essentially talk about those sensors which do not come in direct contact with the food matrix. That's one. And second, we are talking about food nanopackages in which the materials are embedded inside the conventional material. So the nanomaterial does not come in direct contact with the food. Why we talk about out-of-food applications in particular is because there is always a concern about the leach of nanoparticles inside the food. The FDA classifies the food nanoparticles into 2 broad categories. One is in the range of 1 to 100 nanometers, but beyond that there is also one more range which is from 101 to 1000 nanometers. Conventionally, when we talk about nanomaterials, they only fall in the range of 1 to 100 nanometers. But it's only in the food applications that this definition has been extended to 1000 nanometers, which means that when we are altering the size, the natural size of a biomolecule into a nano range to alter its physical properties or to alter its chemical properties, we are also looking at not only dissolution of those particles, but we also have to look at the safety of those particles. The out-of-food applications, such as those in food nanopackages, prevent those kind of concerns of toxicity because the material, the nanomaterial, is not in contact with the food directly. It is only in contact with the food package. It is, let's say, we have two layers of packages of conventional, and within it we have layered it with a nanomaterial. That nanomaterial, let's say, is enhancing or preventing the barrier of oxygen. So that food material, that layer of nanomaterial does not come in direct contact with the food. So therefore, we will talk about in-food applications as well as out-of-food applications. We have just understood the various applications of nanotechnology in the food sector, vis-a-vis within the food and outside of food. In this module particularly, we will be talking about applications of nanotechnology, particularly in food safety nanosensors, nano formulations, and active packaging films. When we talk about food safety nanosensors, we're talking about nanomaterial-enabled nanosensors, which have been developed for enhancing the sensitivity of detection of certain pathogens and contaminants which are present in food. Nano formulations are being developed extensively for enhancing the value of food vis-a-vis the use of hydrophobic nutraceutics in aqueous solutions. Active packaging films understand the use of active packages, smart packages, as well as intelligent packages in nano packaging films. One of the first applications that we are going to begin to understand of nanotechnology in food is the food safety biosensors. And the very reason we talk about food safety biosensors is because we have to understand food safety. Food is a fundamental consumption of every human or every animal. Therefore, when we talk about food safety, we are not only talking about food safety of humans, we are also talking about food safety for the animals. One specific concern that the WHO has towards food safety is they estimate that every year 1/3 of the population falls ill because of contamination, because of consumption of contaminated food. How does this food get contaminated? We have had incidences, international incidences of food poisonings, of food infections. We would understand food poisonings and food infections when they happen on a large scale. The food has to be withdrawn from the market, which means it incurs huge losses for the industry. So it is always incumbent to study about food safety biosensors to know how the food safety assessments are to be done. The food safety concerns are broadly classified into two categories. One is the microbiological risks. This is when we talk about highly perishable foods like milk, meats, poultry, or processed foods such as cheese or other dairy fruits or fruit juices. These kind of foods are highly perishable foods and they fall under the category which is more liable towards spoilage. Therefore, one kind of spoilage that particularly occurs in these kind of foods is the microbiological spoilage, which is mainly caused by Salmonella, Listeria, Campylobacter, or Streptococcus species. The other category of food spoilage is the chemical risks which are involved. The chemical risks will include certain food contaminants, certain additives, food contact materials, and fraudulent adulterants. When we talk about food contaminants, they could be metal toxicity, such as consumption of fish which was contaminated with mercury. Mercury toxicity is a very common toxicity amidst fish because of mercurial poisoning in the rivers. The metal toxicity other than mercury is arsenic toxicity. There is a huge incidence of arsenic toxicity which is being portrayed nowadays. Happens in certain rivulets, where in those rivulets, if there are certain microflora, if there are certain animals of consumption or fish which have consumed that kind of water, there is every possibility of that metal toxicity to pass on through the food chain into the humans. Therefore, it is very important to develop sensors or techniques which are able to sense these kind of contaminants. Additives such as antibiotics are a huge concern. A lot of the food gets rejected in the international market because of presence of antibiotic residues, and antibiotic residue detection becomes one other arena of biosensor detection. Fraudulent chemicals are a concern in the community because a lot of fraudulent chemicals, in particular urea in milk and also melamine in milk, have been added merely for mere commercial gains. It is very unfortunate that this is happening, but as a scientific community, it is our responsibility to develop techniques which will be able to detect these kind of fraudulent chemicals. The table that is shown here enlists the various classes of contaminants and pathogens which are present or commonly found in foods. Mycotoxins, amidst them aspergillus toxins, which are ochratoxins, and fusarium toxins, which are fumonisins, are very prevalent in cereals such as wheat, corn, and rice. Other foods in which it is prevalent are wine and fruits. Fungicides. Kanamycin is one fungicide which is found very commonly in meats and poultry. Other classes of it are also found in wheat-based foods. Antibiotic residues are a big concern when export of foods is happening cross borders. Antibiotic residues are mostly found in meats, poultry, and milk and honey products. Pathogens are a key concern in detection because they owe to food poisonings and food infections. Therefore, it is very important to develop techniques which allow faster and rapid and highly sensitive detection of these pathogens. The most commonly occurring pathogens in foods are Salmonella in eggs, meats, and poultry, Campylobacter in milk and poultry, and Listeria in milk and cheese. Looking at the presence of these toxicants and pathogens in foods, it is important for food technologists to develop techniques which allow the specific detection of these analytes. Therefore, the arena of food analytics. The objectives of food analytics are highly specific detection in which there is no crosstalk. When we say no crosstalk, it particularly means that if I'm trying to detect Salmonella, I should not be detecting Listeria in between. The sensitive detection should be highly sensitive, which also means that we should be able to detect these analytes at very low concentrations, which is called ultra-sensitive detection. This is so because if we have even minute quantities of antibiotics, we should be able to detect them at a very, very small concentration. The objective of rapid detection in biosensors or sensors talks about detection in terms of seconds or minutes, vis-a-vis hours and days. Typically, when we talk about pathogen detection, it takes 24 to 36 hours. But when we are looking at new age sensing techniques, we are looking at detection which ranges from seconds to minutes. There are certain challenges which are posed when we talk about ultra-sensitive detections, when we talk about no crosstalk detections. What in particular are these challenges? When we develop biosensors, we have to overcome challenges of those which are posed by conventional techniques. That is the very reason that we start developing alternative techniques. The conventional techniques are not able to detect certain analytes at very low concentrations, and it is important for certain analytes to be detected at concentrations before which they become toxic. These concentrations are called ultrasensitive concentrations. It is therefore incumbent to develop biosensors which are able to detect these analytes at very low concentrations. When we mean very low concentrations, we should be able to detect them before they become toxic with a high degree of accuracy. This is the first challenge which is posed when food safety biosensors are to be developed. The second challenge posed is crosstalk. In conventional techniques, when we are trying to analyze a certain kind of analyte, there is every possibility that analyte B or analyte C may also be present in the matrix because food is a complex matrix. So the presence of analyte B and analyte C will in all probability interfere with the sensing of analyte A. Therefore, we have to develop techniques which are aimed only at development of analyte A or sensing of analyte A, and not allow the analyte B or analyte E as crosstalks to come in or interfere in the sensing. The third kind of challenge of conventional techniques is the conventional techniques need very extensive sample pretreatments. Let us say, for example, I want to detect an antibiotic in a food. So what will be the need is I take that food, I first extract that antibiotic and then put it to some colorimetric reaction or some biophysical method, and then only I'll be able to detect the antibiotic. This has to be overcome because this is a time-consuming process. We need to develop techniques which are nearly on-site, which would mean I do not need extensive techniques to separate that antibiotic. I may just be possible to dip the sensor into that food and it should give me an output signal. Sounds challenging, but this is how slowly and gradually as scientists we are working towards it, and it is possible too. The one more challenge which is posed particularly in pathogen detection is the time of analysis. Conventionally, when we detect pathogens, which are mostly bacteria, it takes minimum 24 to 36 and up to 48 hours for the detection because we need to culture the bacteria and we need to read the colonies on the plate. This is the challenge which is posed for detection of pathogens. Biosensors are aimed to develop techniques, biosensing techniques, which are able to detect pathogens in very small times, which means rapid detection. In the course of this module, we will see certain examples in which pathogen detections have been done in merely 1.5 hours, vis-a-vis 24, 36, or 48 hours. So these are the challenges which are posed, and we have to overcome these challenges to develop advanced techniques of detection using nanomaterials. Food safety nanobiosensors. Before we talk about nanobiosensors, let us see what are biosensors. Biosensor is an analytical technique which uses two components, the biological component in close conjunction with a transducer. The biological component comprises enzymes, whole cells, antibodies, or aptamers. If I give you an example, enzymes would be, let's say if you want to detect glucose, the glucose oxidase enzyme will be the most compatible enzyme to use. Since we're talking about biosensors, it is important that the biological component is in close combination with the transducer. And the two most commonly used transducers in biosensors are electrochemical and optical. Since we're talking about biosensors, it is important that the biological component of the biosensor is in close communication with the transducer. There are different classes of transducers which are used in biosensors. These are electrochemical, thermometric, wherein there is change in temperature which is detected, colorimetric, if there is a change in color which is visible to the naked eye, piezoelectric and surface acoustic wave transducers, which are typically physical transducers which detect a change in mass or change in the oscillation frequency of the sensor. Optical biosensors, which will talk about fluorescence sensing, surface enhanced resonance sensing, or fluorescence quenching, and field effect transistors, in which there can be the possibility of miniaturization of a device. The most successful kind of transducers which have been developed are the electrochemical and the optical transducers. In subsequent modules, we will take these separately. In this, we are looking at a milk urea biosensor. Urea is one of the fraudulent adulterants which is added to milk, therefore its detection is important. If you look at the second column of the table, you will look at 23 minutes, and on its right you will see 8 minutes. 23 minutes is the response time which was received by microbial based biosensing, vis-a-vis 8 minutes which was a response received by enzyme based biosensors, depicting thereby that in this case the enzyme based biosensor was a better model. If you look at the last column, we have used an enzyme-based biosensor, but it is impregnated with nanoparticles. And looking at the time of detection has come down significantly to 20 seconds. This is yet another example of a nanosensor for detection of small organic molecules such as melamine. Melamine is yet another fraudulent chemical which is used in milk for enhancing its protein concentration. It gives a false concentration of proteins. In this work, scientists have used functionalized gold nanoparticles as detectors. When they come in contact with melamine, their color changes from red to blue. This is yet another example of a visual detection of melamine in raw milk by label-free silver nanoparticles. The difference in this work is that the nanoparticles are label-free, which means that they have not been ascertained or they have not been functionalized with any groups. So the yellow colored dots that we see are silver nanoparticles, which when they come in contact with melamine, which is the structure shown in the middle, they agglomerate and they change their color to pink. The TEM images of the same are depicting the same. The image which is labeled B shows the silver nanoparticles without the presence of melamine, and C shows the presence of silver nanoparticles in the presence of melamine, which is leading to agglomeration. This work is a typical example of how the use of nanoparticles in matrices can enhance the response of biosensors. One such biosensor developed was a biosensor for superoxide anion radicals, which is a reactive oxygen species. By the use of superoxide dismutase enzyme immobilized on a carbon nanotube in a polypyrrole matrix. Polypyrrole itself is an electrically conducting matrix. However, when carbon nanotubes are added to that matrix, it enhances the electrical conductivity of the biosensing element. What is depicted in the figures below is, on the right, we see a dotted line, which is an almost flat sort of curve. Whereas on the left, if you look at the dotted line, we will see at a position at point A, where there is an appearance of a distinctive peak. Similarly, there is an appearance of a distinctive peak at position B on the left figure. These appearances of distinctive peaks are only happening after the addition of carbon nanotubes in the matrix. This figure, which is a cyclic voltammogram of a response, clearly depicts what was explained earlier, that when carbon nanotubes are embedded into a matrix, it enhances the response. What is seen clearly is a very sharp peak, an oxidation peak which appears above, as well as a reduction peak which appears below, both of which are very sharp and clear. This is an example of pesticide detection, again using carbon nanotubes with polyaniline as an immobilization matrix. In this work, pesticides were detected in fruits and vegetables. What is important to note here is the sensitivity of detection, which is as low as 1.4 micromolars and a short detection time of only 10 minutes. This work shows the detection of a pathogen Salmonella by using gold nanoparticles as well as magnetic nanoparticles. What is important in this work is a response time as short as 1.3 hours vis-a-vis conventional techniques which could take from 24 to 36 hours. The second application that we're going to talk about is nanoformulations and how these nanoformulations are used as vehicles for value addition of foods. What do we mean by value addition? Most of the nutraceuticals are hydrophobic in nature, which means that they cannot be utilized in their full capacity by the human body, because human body is 70% aqueous in nature. So how can we convert these hydrophobic molecules into how can we lend hydrophilicity to these hydrophobic molecules so that when they are consumed, they become more bioavailable to the system? So we will study how nanoformulations are used to achieve this objective. Since we have shifted attention of application of nanotechnology to yet another field of nanoformulations, one of the examples that I would like to detail is the use of nutraceutics in foods. Nutraceutics are naturally healing molecules which are present in food. They have therapeutic properties, but they have certain limitations too. One of those limitations is that most of these nutraceutics are hydrophobic in nature, which means that they are not aqueous soluble. This poor aqueous solubility renders low bioavailability to these nutraceutics. What does this mean? This means that if we are consuming these nutraceutics, only a very small percentage of it will be bioavailable or available to the body, because our body is 70% aqueous. Therefore, nanotechnological applications have looked into increasing the aqueous solubility of these kind of formulations, and the most popular kind of formulation in foods is nanoemulsion. Nanoemulsions, as also liposomes, are lipid-based nanoformulations which have been used or developed successfully with enhanced bioavailability of these nutraceutical compounds. In particular, nanoemulsions have been prepared for beta-carotene, omega-3 fatty acids, and tocopherols. A company by the name of Neutralise has developed novel carriers for nutraceuticals, which is to be incorporated in food systems and cosmetic nanoformulations. What happens with these is the bioavailability of the product is increased by a technology called nano-sized self-assembled liquid structures, the NSSL. This company has developed these vehicles and used them for increasing the bioavailability of coenzyme Q10, luteins, lycopenes, phytosterols, and vitamin D, all of which are classes of nutraceutical compounds. Shown in this figure is value addition of food by the use of nanoemulsions of beta-carotene. Beta-carotene is a nutraceutic with provitamin A capacity. Otherwise, beta-carotene molecule is a hydrophobic molecule. It cannot be dissolved in aqueous systems. Therefore, we developed nanoformulations as nanoemulsions of beta-carotene and used it for value addition of aqueous as well as fat-based foods. The upper portion of the figure shows how these nanoemulsions, beta-carotene nanoemulsions, have been used for adding value to butter, whereas the lower portion shows how the beta-carotene nanoemulsions have been added to lychee juice. Typically, had we added a salt of beta-carotene to these formulations, it would have not got dissolved because of its hydrophobic character. Yet another application of nanoemulsions is their antibiotic activity or antimicrobial activity. It has been shown and it has been reported by workers that nanoemulsions are very effective against a great variety of food pathogens, particularly gram-negative bacteria. Shown in this slide on the left is treated, whereas on the right is shown an untreated plate showing the growth of Salmonella typhimurium. It is clear from this picture, a clear plate shows the antibiotic character of the nanoemulsion. Beta-carotene, we have established, is a nutraceutical compound. So is curcumin. Curcumin is a bioactive character which is present in turmeric. Both of these are again hydrophobic in nature, which is attributed to their structure. The figure on the left shows nanodispersions of beta-carotene in aqueous solutions of chitosan-based amphiphiles. You can see very clearly the different colors of the beta-carotene solutions, the nanodispersions with the increase in color. On the right are curcumin flakes, which are not in a nanoformulation, vis-a-vis curcumin flakes which are in a nanoformulation, which is seen to be completely soluble in water. One of the final applications that we will introduce you in this module is the food nanopackages. As briefly told before, the nanopackages are being developed to enhance the properties of conventional packages. These nanopackages are broadly classified into three categories: improved packages, active packages, and smart packages. Improved packages means where you have improved the mechanical properties as well as the barrier properties of the material. Active packages would include those packages wherein some active materials such as antimicrobials or antioxidants have been added, which enhance the shelf life of the food. Smart materials are essentially those which have indicators such as nanomaterials in which certain indicators have been put, which will change color when the physical state of the food has changed. During the course, we will see certain specific examples of all these three kinds of packages. Talking about applications of nanotechnology in food packaging, what is shown here is how these applications are used in enhancing the character of food packaging films. with which the food can remain fresh for longer. In the image shown on the right is impregnated and oxygen absorbing layer within the food nano package, which helps in increasing the barrier properties of the film. By increasing these barrier properties of the film, the shelf stability of foods can be enhanced. Though still in its conceptual stages, but what's shown here is a milk pack that changes color every day as the expiry date gets closer. What's shown here is a work which has been done by a university in Lithuania in their process of developing antioxidant films. In this, essential clove oil, which has antioxidative character, was developed. But a step ahead of that was that silver nanoparticles were also added to this nano formulation to enhance the tensile strength of this packaging film. When we talk about improved packages, we are talking about improved characters in terms of mechanical strength as well as enhancing the barrier properties of these packaging films. When we talk about barrier properties, we are trying to limit the transport of any gases through the packaging films. The figure on the left shows a linear path for oxygen, whereas the figure on the right shows a sort of tedious path which the oxygen molecule has to take, which improves the character of a food packaging film because it takes longer time for the oxygen to permeate inside the film. Therefore, the diffusing gas molecules do not pass perpendicularly, whereas these diffusing molecules navigate around nanocomposites, increasing the mean gas diffusion length and thus the shelf life of foods. Though we have discussed all the opportunities that food nanotechnology opens for us, such as food safety nanobiosensors, nano formulations as vehicles for value addition in foods, as well as packaging materials which are nano-enabled for food packaging films, yet there are certain challenges which are posed. One of them is the safety issue. The safety of nanoparticles needs to be ascertained, particularly when they have to be used directly in food applications. Therefore, we talk about regulatory compliance. The FDA classifies nanoparticles to be used in food in two broad categories, one from 1 to 100 nanometers and the other from 101 to 1000 nanometers. Therefore, typically what we would think of a nanoparticle in the dimension of only 100 nanometers, but in food technology or in food applications, this dimension enhances to 1000 nanometers. Therefore, when we develop processes or when we develop products which are enabling the use of nanoparticles, we need to see that whether these fall in this category and secondly, whether these are safe. Third, the affordability of these nano-enabled applications. Though we are talking about very distinctive applications of nanotechnology, yet we do not have to forget that while developing these applications and processes, these should also be affordable. We would conclude now, and we've understood the various applications of nanotechnology, in particular the food safety biosensors, the nano formulations, as well as the food nano packages. However, before we take up any of these applications on a commercial scale, it is important to keep the FDA regulations in mind. Other than that, once we are able to do that, these applications will all open up a whole new generation of products and processes in the food sector.